welcome everybody to a rather special and unusual event. This is Brain Awareness Week, and it's got a special title, Your Brain is on Our Mind. So consider your brain part of everybody's interest at the moment. Mine certainly is. Welcome to guests and friends of QBI, and thank you for coming to this event to learn more about what our exciting and unusual colleagues are doing. To begin, I acknowledge the Turrbal and Yagara people as traditional owners and their custodianship of the lands where we meet today. On behalf of the University of Queensland, the QBI, and all present, I pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. The way we're going to do today is I will make a short introduction, and then our three specialists will talk a little bit about themselves and their work, and then we're going to have an extended panel discussion, which I will facilitate, but like a good MC will keep out of the way of. And they will discuss various important issues relating to their current work and how the bits of research interrelate in the QBI. And then we will have questions from the audience. There will be a roving microphone, and if you put up your hand, someone will find you, and we'll be able to record your contribution. This whole thing, by the way, is being recorded. It's going to be part of the website of the QBI, and so we are going to be immortal in one way or another. My name is Rowley Sussex. A number of you know me already. I'm pleased to see a number of very familiar faces here. I'm possibly here because I've been broadcasting on language for the ABC for 26 years, but possibly also because I'm a linguist. And language, for me, is one of the most astonishing products of the human brain. There are still about 6,900 languages extant in the world, which is an extraordinary achievement of creativity, if you like. And I think I look at this with wonderment at the way in which not only people can have one and several, and in one case, 92 languages. There's a man from the US military who was supposed to get around in 92 languages. I don't know how he had room for breakfast, but never mind. But part of my relevance here is as a linguist, but also I've got a big project on pain and communication, how people talk to their doctor about pain. And of course, pain is felt not necessarily in your toe, but actually in your brain, and we'll have some more about that as well. The history of this place is quite unusual and special. In October 2002, Professor John Hay, who was then Vice-Chancellor of the University of Queensland, Chuck Feeney from Atlantic Philanthropies, and Professor Perry Bartlett from Melbourne got together and had an epiphanous moment. They decided to have a really, really big and radical idea, which was a brain institute. They went to see Peter Beattie, then Premier, and the Queensland government came on board, and by the end of 2002, basically the funding was in place. The institute was approved in 2003, and the 5 founding members began, and there are 4 of those still associated with the institute, which shows you how well they manage their people. They worked in the McGregor building, and then the Ritchie building, and then in 2007, in that splendid special building at UQ, and if you haven't visited it, you should. It's something really quite unusual. Today they have, from a standing start 20 years ago, 350 researchers. It's the biggest in Australia, including 110 PhDs and 100 postdocs. And they have people from more than 40 countries around the world. This is, if you like, a beautiful microcosm of what Australia ought to be like, an international meeting place of ideas. And this place is among the world leaders in brain research. They've got 5 big focuses. I won't say too much about them because they will, but brain development and plasticity, cognition and behavior, aging and dementia, brain injury, and mental health. But they also, if you've been on their website, you will know this, have a very strong commitment to getting their knowledge out there into the public sphere and making it understandable to people like you and me, who may be interested but not specialist, and who want to know what is going on up here. And your brain, well, there's about 1.4 kilograms of it. And it's got weird things in it, like the amygdala, which is the Greek word for an almond. There's something in there that looks like one. And it's got a hippocampus, The Greek word hippos means a horse and campus means a sea monster. And so your hippocampus, which is very important, as you will hear from our colleagues, actually looks like a sea monster, but you need it very, very badly. Don't be rude about it. Your brain contains 100 billion brain cells or neurons linked by trillions of connections called synapses. And a piece of brain the size of a grain of sand has 100,000 neurons and 1 billion synapses. It is the most incredibly compressed bit of physiology I've ever known about. And a child's brain is a third the size of the adult's, and it grows at 1% a day. The next time your child deletes all your files from your iPhone, be grateful that a bit of neuroplasticity has taken place and the child has learned something invaluable about digital objects. All right, I'm going to get out of the way. Our three speakers today are Professor Pankaj Shah, who is director of the QBI, Professor Jason Mattingly, who specializes in attention, and Dr. Janisa Javeri, anxiety and depression. They're going to say a little bit about themselves and their work before we have the first discussion. Pankaj.
Thanks, Rolly. So I'm Pankaj. I'm the director of QBI, and I started life as a medic, trained in medicine at University of New South Wales, and pretty much finished my medical degree and got interested in the science, and realized at that time that really, while we were learning a lot about kidneys and lungs and knew how to treat a lot of the disorders, there was very little available in terms of people with neurological disorders. And really at the time, my interest was mostly neurological disorders, and decided to do a PhD in physiology and studied proteins at the time and single molecules, and then went on to stay in science ever since then, and have become interested in the amygdala, which is this bit of the brain which is called the almond, and it processes emotions in our brains. So my laboratory basically works on the connections of the amygdala and how emotions, particularly fear, come around. And we also work with people with movement disorders. My first love was Parkinson's disease, essential tremor, which we use electrical techniques to stimulate the brain and understand what's happening in these people. So I've been in the QBI since the start. I was one of the founding members, and have been there 20 years now.
Next, Professor Jason Mattingly.
Thanks, Rolly. So my background is in psychology. I did a Bachelor of Science degree at Monash University. When I finished that degree, I thought, oh, actually, I'd like to help people, but I was very interested in the brain, so I decided to study neuropsychology, which is the study of people who have brain injuries of different sorts. So I went to Melbourne University and did a master's degree there. And I enjoyed that experience. I loved working with people and in the clinical environment, but also was really passionate about research, much like what Pankaj said, and so ended up going back to Monash and doing a PhD. And my PhD was in stroke, and particularly looking at the kind of sensory and perceptual problems that people who have had a stroke can experience, and looking at patterns of recovery after stroke. When I finished my PhD, I went on a fellowship over to Cambridge University. I was there for several years. I was a fellow of King's College, and I worked in one of the Medical Research Council units over there, the Cognition and Brain Sciences unit. And I continued my work on stroke, but also got very interested in normal, healthy brain function as well. Came back to Australia. I was a faculty member at Melbourne University for several years in the School of Psychological Sciences. And then Perry Bartlett, who was the founding director of QBI, invited me up to Queensland to give a talk. And little did I know that actually he was trying to headhunt me. So I gave the talk, and when I came out from the talk, his secretary passed me a huge dossier, and it was full of real estate prices and schools. And I thought, am I really here just to give a talk? Anyway, one thing led to another, and I couldn't turn down the offer of moving to UQ and QBI. I joined in 2007, and I run a large research group. Our interests are really focused on, we start with behaviour and function, and we then ask, what are the brain processes that regulate those functions?
Thank you. As you see, the QBI takes care of its people, including real estate. Janisa Javeri.
Yes, so I'm a developmental biologist. I did my training, master's and PhD back in India, in Mumbai. And during my PhD, I was investigating how the fruit flies, Drosophila, actually develop their sense of smell, how their neurons in their antenna actually connect up in the brain. And that was quite a fascinating journey. And then I was trying to look for a postdoc position overseas. And at that time, my husband said, I'm only moving to a country which speaks English and plays cricket. So here I am. I came here on an international scholarship, the Human Frontiers Science Program. Perry Bartlett, again, recruited me. And I started working with him, fascinated by this idea of regenerating brain cells in the adult. Something that was at that time still considered a dogma, because there was a huge belief in the last century that your brain is immutable, hardwired. You cannot really regenerate new cells, unlike some of the other organs like skin and blood. But that dogma was overturned, and some of the seminal discoveries that Perry made, I was really interested in, and that's where I did my postdoc. And since then, I have continued working on really this regenerative capacity of the adult brain, trying to find ways and look for mechanism by which the stem cells in the brain actually produce these neurons, how they make the right connections in the circuit, and ultimately trying to understand how they contribute to our cognitive and mood functions. And particularly, my laboratory has focused around looking at the role of stress, how stress actually impacts this process, and that may lead to some aspects of anxiety and depression.
That'll give you some idea of the enthusiasm. And when you go near the QBI, you could just about feel the buzz. This place is alive. And so the first question to all of the three panelists is, what fascinates you individually about the brain? Who would like to go first?
I'm happy to jump in.
Jump.
All right. I think I was talking to Denisha about this before, and what fascinates me is if you look across all of the animals that have a brain, the basic building blocks are the same. The nerve cells, the neurons, connected to one another into circuits that subserve functions and behaviours. But look at the huge repertoire of different behaviours that we see across animal species. So to me, there's something fundamental about those basic building blocks and how they're connected that can support all kinds of different functions. If I were to focus specifically on humans, my interest, I would say I'm very interested in affect and emotions. There's a lot of talk these days around things like intelligent systems, artificial intelligence. What we don't really fully understand is how we might imbue those artificial systems with things like emotions. And we may come back to talk more about that later, but I think they're the things that really fascinate me and big unanswered questions at the moment.
What fascinates you, Denisha?
I like to view this blob that we have between our ears as a really fascinating rainforest. It's rich in diversity, incredibly beautiful, and has more cell types than perhaps any other organ in the body. And with modern neuroscience tools, the fact that actually we can visualize these cells, see them in action, learn about how these cells, these neurons, talk with each other and communicate, and what function they serve at an incredible detail, almost at a single cell resolution, I think is something that I'm really, really excited about.
Pankaj.
So I guess grew up as a technical geek. In high school, very interested in computers and programming, and did quite a lot of it. And when it came to thinking about brains and the kinds of things that the brain does, my original, a lot of programming that I did was doing very simple things, in retrospect. But as soon as it came to thinking about brains, about how it is that information gets represented, and how it is that you have thoughts, or what kind of thing a memory might be, considering how many memories get stored. And when things go wrong, they get to go pretty wrong. And how is it that something like that might happen in a bit of mush in your brain? I think that's really pretty amazing, that something is going on there. It's electrical, and it does all the things that we do. So I just find the whole operation of this device pretty amazing, and trying to understand what is it, how it functions, and what might go wrong.
Lovely. I can remember when I first started reading about psycholinguistics that one of the standard mantras was that the brain is fundamentally set. It's got a structure, and that's it for life. And yet, is it possible to rewire our brains? Who wants to have a go at that first? Okay, go for it.
So yes, it is indeed possible to rewire. And I bet many people here, or maybe not many people, but you've heard of people who've had a brain injury, and for a while things go awry and they don't work very well, and just an area Jason has worked in, particularly following stroke. And often, you don't always recover, but many times you do recover completely. And how the recovery happens is by rewiring. And this is not always just new growth, but it's connections which are latent, get turned on. So the brain has an incredible capacity to fix itself and rewire. And one of the stories I like telling my students is, on a Friday night, you could go out and get completely hammered. It's not a good thing to do, but you still find your way home. Okay, and how does that happen? And that's the short-term plasticity that your brain does. It's the manages to find those connections, despite the fact if you pour a cup of coffee on the edge of your computer, that's just had it. It's not going to come out of it. So it's a very, very interesting place, which is very plastic, very malleable, and is very, very able to adapt.
Jason?
Well, I was going to, just to elaborate on that, I think you raised an interesting point. I always think about these two parts of brain function. One is the structure, the cells and the wiring that Pankaj talked about. And as he said, any kind of damage, things like a stroke or a head injury, there's a capacity for regenerating the wiring, but there's also the functional side. And when we talk about functions in brain science, we're talking really about the synapses, the little gaps between the cells where neurotransmitters are passed between them, and that allows communication. And we think a lot of learning, probably all learning, is focused around what happens at the synapse, remodeling of those connections. If you come from a computer science background, you would think about changing the weights of the connections between one area or another. And so I think when we think about plasticity, it's good to think about the structural changes, the cellular and circuit changes, but also changes at these little gaps between the nerve cells.
Okay, structure changes and function changes.
Right.
Danisha?
That's it. I think it's, and one of the extreme form of this plasticity is something that I alluded to earlier, which is adding new cells into the circuit. So you can imagine that already this circuit is sort of plastic, but now when you are adding new cells, you have to make some space for these cells, and they have to make the right connections. So they have to talk to the right neuron in the right way. And that itself is, a big challenge, and it's something that happens with a lot of learning. It happens with environmental enrichment. It actually happens with physical exercise as well. So there you go, in terms of you can have structural, functional changes, you can have addition of new cells into the circuit. So it's quite a disruptive process.
Welcome to the disruptive head. Okay, now this is interesting stuff. As a linguist, I'm interested in people who might suffer damage to the left frontal lobe. And yet the brain is able to rework things? How does that happen?
I could try and field that one. So you're right, Rolly. Many people who have a stroke, particularly a stroke affecting the left side of the brain, which we often think of as the language hemisphere, if you have a lesion that's in just the right place in the frontal area, then people lose the ability to express language. And so they have sort of faltering or hesitant speech. They forget about how to use grammatical function words, for example. Depending on where the lesion is, you can have people who can't comprehend language anymore, but they can still express things. After those kinds of insults to the system, as Pankaj mentioned, there's often rewiring and recovery. And that can happen spontaneously, but we have a whole field, speech therapy, speech pathology, where experts in language try and retrain the system. It's laborious, it's difficult, but there are often very, very good outcomes.
And some people are successful and some are not?
Well, it partly depends on, there are a range of factors. The size of the stroke, the age of the person. Often insults to the brain that happen very early on in life, there's much more opportunity for the system to change and adapt, to rewire. The older we are, the older we get, the more difficult it is for the system to recover. That's not to say that there isn't recovery, but it's just more difficult.
I'm trying to learn Chinese, and I can tell you that's true. Danisha, you talked about structure and function. The brain at 1.4 kilograms is about 2% of our body weight, and yet it uses about 20% of our energy. How does that work?
It's an incredibly energy expensive organ. I mean, I think the fact that, you know, the brain never switches off. I mean, we can rest, we can sleep, but the brain is not off. It's still going on. And the myth that, you know, we only use 10% of our brain and 90% is there somehow, we can unlock it, is not really true. In fact, studies using magnetic resonance imaging have shown clearly that even at rest, a lot of our brain areas are actually, in fact, active. And as Rolly had alluded earlier, we have 100 billion neurons, probably 100 trillion connections. So just to maintain them, there is going to be a lot of energy that's going to be required. And the majority of the energy is required at these points of contacts between neurons at synapse, because that's where the information exchange happens. So, you know, there's a flow of ions in that space. That's our electrical sort of messaging. And just to keep that flow going, there has to be some pumps which are functioning at that level. And to keep those pumps going, we need a lot of energy. So it's not really surprising that we use 20% of the energy. And certainly it's not one of those devices where we can put a 5-star rating.
Lovely. London taxi drivers are supposed to have differently structured brains after learning to be London taxi drivers. Basically, they spend months and months and months riding around the city on little scooters, learning where all the streets are. Nowadays, by the way, I suspect they use a GPS and no one has this benefit anymore. But there used to be an important period when they were learning, and until they qualified as a decent taxi driver in London, they had to know where everything was. How does that affect the structure of the brain? Pankaj, would you like to? Is this a hippocampus?
Yeah, I mean, learning is something that happens. So every time you're looking around, and in fact, these taxi drivers, you know, they used to do a test where they walked around, they were trying to learn the city. And each time you learn, what happens, the theory is that the strength of these connections changes, and there's growth of some of these connections as well. And all of it starts in this bit of the brain called the hippocampus. So these taxi drivers, they had a little bit bigger. The hippocampus actually got a little bit bigger. And some of the kinds of experiments that Dementia was talking about during this learning, the thought is that new neurons get even born in the hippocampus during this learning. So they take a bit more room. So there's a bit of growth as well. So the idea is that there's a huge amount of plasticity and growth going on in these people. And that's why their brains change.
Wow.
I think that's where, sorry, I was just going to add that in the hippocampus are these place cells and grid cells, which actually represent our surrounding information. So these cells actually fire only when you are at a particular place in a spatial location. So there is this whole map in the hippocampus about our spatial sort of location, and that perhaps changes when you have to use a lot more of that navigation aspect, I guess.
I was going to say that when that study came out, it caused huge waves, this idea that your brain gets bigger when you learn a spatial map. And scientists were rightly skeptical initially. They thought, well, maybe there's a cause and effect problem here. Maybe the people who decide to become London taxi drivers have a big hippocampus to begin with, right? So they kind of self-select it. They know they have a good memory from the very beginning, and they're the ones that become successful. But there's been a whole bunch of follow-up studies showing that if you take individuals at the very beginning, before they've done any spatial knowledge learning whatsoever, do an MRI scan, look at their hippocampus, they're the same size, and then test them again 4 years later, the ones who learnt to be taxi drivers and the ones who didn't. The hippocampus has grown in that time.
Does that apply if I learn the tuba?
Learning a musical instrument is another great example of how you can induce plasticity in the brain, indeed.
Okay. Right, because linguists often know that there are three things which go together: mathematics, language, and music. I got 2 out of 3. I won't tell you which one I missed. Pankaj, how do we control movement?
So, very interesting, right? So this is one of the things we all do, and the brain's very good at it. Very fast. If you think about, cricket players, this ball is coming at you at 100 kilometers an hour, and yet you manage to react very quickly. So in the back of your brain, you have your second brain called the cerebellum, And the cerebellum, in fact, has as many cells as the rest of your brain put together. So it's a tiny bit of thing, as these are packed in there, and it's the cerebellum that controls movement by and large. A lot of the actually where you put your arms and legs, exactly how fast you move them, and when you stop is all run by the cerebellum. But the planning is done by your motor cortex, which is a bit of your brain up here. So the way the movement is controlled is there's a starting point where you decide to move, and that's what's called premotor planning. So at some point you have to plan that you're going to move from here to there, or you're going to catch this ball or hit this ball, or whatever you're going to do. That message then goes to your motor cortex, which then decides exactly how you're going to move, and then that goes to the cerebellum, which then drives all your muscles at just the right speeds. So it's a pretty interesting organ, and we exactly don't know what it's actually doing. We have some vague ideas, but not really that much.
It does all that without us having to think about it.
Yeah.
Precisely.
Yeah.
Very fast.
Yeah.
Yeah, the speed at which some of these things happen is really quite phenomenal. Because if you imagine, as you said, that the cricket ball is approaching at 100 miles an hour, you don't have much time to work out what your best defense is. Anyway, okay, what about regulating emotions and behavior, Danisha?
Now, that is a question that I think has captured neuroscientists for probably last multiple decades. It's a question which is still, we are searching for answers. But I can tell that I think, first of all, just trying to understand where these emotions are coded, which part of the brain and how they are coded. Say, for example, as Pankaj said, fear, which is largely the organ, the region which is responsible for controlling that is amygdala. Hunger, we know now that it's the part of the brain, the hypothalamus, which actually codes or regulates some of the feeding behavior. Aggression, love. So I think there are these emotions which now we have come to appreciate that some of these are coded in certain regions. These are sort of specialized regions, and we are just beginning to understand the circuit by which these emotions are coded. And we are able to do a lot of this to tease apart these circuits and which cell types are involved using animal models. I think with humans, we can actually look at the MRIs and look at which brain areas light up when we are feeling emotional, either happy or sad or grieving. But in animals, we can actually also starting to actually tease apart which neurons are doing what. And for example, something that we are looking at is an approach avoidance kind of behavior, which is related to fear. So you can imagine that if you have to be in a novel environment which has some threats, you have to gauge in terms of whether to venture or to hold back. If you are overcautious and you don't really venture, that there may be a price to pay. You may not get your rewards, such as food or finding a mate. And if you are, so this decision-making is kind of very important, and dysfunction in this decision-making really results in anxiety sort of type situation. And what we have found, and this is some of our recent work in the lab, is these new neurons that I talked about in the hippocampus can regulate this approach avoidance behavior. So we can actually selectively regulate their activity and see in real time that the animal now no longer is moving towards the reward and is actually avoiding that. So I think this is where we are beginning to get really good handle on the cell types, the circuit which are controlling some aspects of this emotion, and how that then manifests in behavior.
Okay, I'm interested in memory because I like to learn lots of words. Pankaj, how are memories formed? You mentioned them before. How are they retrieved and managed?
Well, there's a theory. I guess Jason can probably help me here as well. There's a theory about how memories are formed. We talked about all these cells in your brain, all the connections. And the theory goes something like this, that these connections encode information. And when a memory is laid down, in the end, it's information of some sort which gets put down. And there's a three-step process. So there's a learning process in which you recognize something and have to learn it. And then there's a short-term phase, and many of us, when you think about a phone number, there's a short phase in which there's a short-term memory, which lasts very brief time, and then it's forgotten. And then there's a consolidation process, okay, which overnight normally takes time, and sleep's involved in this as well, during which it moves into a different part of the brain and is consolidated and is there forever. And what's supposed to be happening is that at each phase, the strength of these connections changes. Okay? And the final memory that's laid down is a pattern of connections in your brain. So when you recover your memory, and we have to recover it, this pattern of connections somehow gets restarted. Okay? And the connections are distributed through your brain, the long-term memories, and exactly how it is that you manage to start these whole connections up again and then retrieve the memory is not really understood, really. You know, for simple things, we have some ideas about how they might be formed, but for a memory, the kinds of memories that we have, like the memory of, you know, what you had for breakfast this morning, we have really not much idea of how it might be encoded, what the encoding would be, and where it might be.
Because I suppose there's an olfactory input, there's a visual input.
Oh yes, our memories are formed
input,
complete all sensory modalities. You know, every part of our sensation feeds into it. And you know, you can learn things by closing your eyes, but if you have your eyes open, you remember different things.
And it's important also to remember that it's not one system. We often talk about memory as if it's a thing, but it can fractionate in interesting ways, and Pankaj sort of hinted at this. So that, you know, what did you have for lunch yesterday? We would call that episodic memory. It's a memory for the events and episodes of your life. But then there's another kind of memory that psychologists would call semantic memory, and it's really about the facts that you know. So when I say a word like cat or dog, you can create a kind of image in your mind's eye of what a cat or a dog is, and you know a lot about it. But you know, you don't necessarily remember specific episodes with every cat you've ever encountered. So semantic memory is a kind of different system. And then we have the automatic form of memory. Sometimes it's called procedural memory. So your memory for how to ride a bike would be a good example of that. And those systems function pretty independently. We see in cases of pathology that episodic memory can be impaired, but semantic memory and procedural memory can be intact. And all of those individual systems can be affected independently of one another. It just kind of highlights the fact that memory is not one thing, it's many things, and it's a dynamic process. The other thing we often don't appreciate is if you go through the act of remembering something. Maybe a moment ago you thought, what did I have for lunch yesterday? If you do that exercise now, just by recalling that information, you will change the memory. So it comes back to that network that Pankaj talked about. You activate the network again, you're subtly changing the connections in that network. And that's why, for example, in law enforcement, when people ask for eyewitness testimony, they have to be very careful about the way they ask questions, because just by asking witnesses to recall an event, that action in itself changes the nature of the memory.
And some memories can be triggered in ways you don't necessarily anticipate. I was living in England years ago, and I went for a walk down a country lane, and someone was burning eucalypt twigs. And suddenly I was back in Australia. Smells can be very powerful. I don't know why. Okay, is it possible, Pankaj, to stop, reverse, or remediate dementia? Because you were talking about this at the start.
Dementia is a complicated process, and it's not one disease. There's many, many different types of dementia, and they happen for different reasons and have different, I guess, processes that proceed. And first off, if dementia is at the stage where, you know, there is a lot of loss, so a person is very severely affected, we know for a fact that in those people, it's happening from cell death. So cells have actually died. So during the dementia process, it doesn't matter what kind of dementia it is, ultimately you're losing cells in your brain. Once the cells are dead, it's very hard to recover from that. Okay? So recovery from dementia, let's say late stage dementia, is right now, we don't have any good way of thinking how you might do that. Now, is it possible to prevent it? Maybe. You know, we are developing techniques at the QBI, which may be possible to delay it. One of the things that we know about dementia is the biggest predictor for dementia is age. You know, it's not whether you smoke or whether you drink or whether you don't exercise. Age is really the biggest predictor of it. And why some people get it and some people don't, that's really a big question right now. But with early diagnosis, there are definitely ways you can slow it. So that doesn't really answer your question, but that's the kind of place we are in right now. And the research that's going on in QBI right now, what it's trying to do is really delay it rather than cure it.
Pharmacological, behavioral, lifestyle.
All those things,
Yes.
all those things, exactly. There are some pharmacological treatments available right now, which are a little bit effective, but not that effective. And we have some ideas about which things you should change to delay it, but we don't have anything right now that will stop it. Yep.
I want to talk about artificial intelligence because that's been in the news recently. Many of you will have heard about ChatGPT. A friend of mine got on and asked him about me. And was quite pleased to find that I'd made contributions to language and linguistics and so on. But unfortunately, I'd passed away in 2018. I kid you not. I got onto ChatGPT and said, I am, in fact, Roland Sussex, and I'd like to tell you that I haven't passed away. Can you fix your data? And I got what I suppose in digital terms is a groveling apology. We are terribly sorry to have caused you this discomfort, and we promise we will fix it. Someone checked up on me the following day and found that my life expectancy had now got advanced to 2021, when I'd last been seen alive. So, but this stuff is with us, and it's important, and it's pushing all sorts of limits out. Where are we with AI, and what are the good bits, if you like, and possibly the threatening bits of it? Who would like to tackle that?
I'm happy to have a go.
Have a go.
Yeah. I mean, I think developments in AI are amazing and fantastic. We do need to manage them, and we need to bring people, society with us in those developments. One thing that often people don't realize is that the most recent forms of artificial intelligence, things like deep learning algorithms, deep convolutional neural nets, those things are all inspired from brain science. The organization of those algorithms is inspired by the organization of the brain. The idea that the brain divides up, starts with things that are very simple and builds up a kind of more complex picture over time. That's how these algorithms are organized. In terms of fears and concerns, I mean, I think from an ethical and legal point of view, maybe what we have to think about is if we have an autonomous vehicle and that vehicle kills somebody, a pedestrian, who's responsible for that? You know, if you're out and about and suddenly you're arrested because you're suspected of a crime because an algorithm identified your face at the scene of a crime, you know, how happy are we that might happen? So I think from a legal and ethical point of view, we need to be very careful. But these new algorithms, ChatGPT is a good example, are very, very powerful. I think they potentially have a big role to play in helping with learning. At the start of this semester, in my teaching course, undergraduate psychology, I asked ChatGPT to create an entire syllabus for the entire semester, just because I wondered how would it compare with the one that I gave, that I already had created. And it was very similar to the one I'd created. So if it was coming in from scratch as a new lecturer, that would be a really useful tool.
On my right, there is an AI at work. We talked a lot about the physiology of the brain today. Does AI learn anything or pass anything in either direction, from AI to your work and from your work back to AI?
It definitely is going in the direction where the ideas that we're learning about how mammalian systems learn and some non-mammalian ones. Some like reward, for instance, which never used to be there in the early software that was written, is now incorporated. In the direction of us thinking about what the biology is doing, I'll give you one example of it. in which we were working on a particular problem in the amygdala. And then we were trying to think about, we had some experiments we'd done, measuring signals in cells. And we started talking to some artificial intelligence modeling people. And then they created a model which was completely a model. It sort of used some of our data, but their model had predictions which we had not even thought about. So it works in that direction by making predictions. And we did the experiments that their model predicted, and that changed the way we were thinking about how the whole system operated. So it gives us ideas about experiments. And that is a lot of the way the feedback in that direction is going.
But the physical architecture of AI systems is very different from that of the human brain. Do you think
actually,
they might?
They're coming together. They are coming together. Some of the new systems that are being built are called neuromorphic chips, and these are silicon chips, but they have a little bit of the biology built into them, if you like. And the brain-machine interface, I mean, some of you might have heard of brain-machine interfaces, these devices that talk to your brain, those are coming our way as well. They're getting better and better. And so there will be a communication between the two in the future, and this will develop very rapidly.
Wow.
There was a paper published recently, some people might have seen this, where they took one of these neuromorphic chips, so a silicon wafer with real neurons in a solution, and wired it up to a computer game and had it learn the game of Pong. If you're old as I am, you'll remember this game. It's like tennis on two sides, two little paddles and a ball sort of moving between it. But it basically was able to wire itself up to play that game without instructions.
Wow. Okay. I hope you will allow me an indulgent question. I'm doing a lot of work at the moment on pain and how people communicate pain to their doctors, because what you manage to communicate is very important to the diagnosis. Danisha, how does the brain perceive pain?
Oh, I think Pankaj will be a better person to answer that question.
So look, I'll have a look. The pain is very interesting. I mean, all of us have felt pain, and what pain is, in the sense of your feeling of it, is a very personal thing. And somebody says, "Look, I've got a headache." Well, they've got a headache. That's the end of the story, basically. So why do we get pain? Well, there's two systems. One is that we definitely have pain receptors, some that respond to heat, some that respond to cold, and others to vibrations. And this system feeds into your spinal cord and goes to this part of your brain called the thalamus, which is a way station, and then ends up in different parts of the brain. Now, it follows the very, very similar pathways to the way sensations go. Okay? So if you touch a hot object, the sensation of touching it, those signals follow very similar pathways to when you touch a hot object, that stays. So there are pain receptors. So exactly how it becomes a pain is, again, not very clear. And of course, it depends on your emotional state. The same sort of injury that you have on a sporting field, where you bash your ankle or you twist your ankle and you feel like, well, I have to score this goal, you keep running. It's very different to when you're just walking down the road and you bend your ankle and you collapse from the pain. You can choose to ignore that pain when you're playing a sport in the middle of a fantastic goal. So it's driven by emotion and your sensory state. So pain is one of those sensations which varies from people to people, and how it's processed appears to be regulated by your whole emotional state, basically.
I think that's an important point that you raised, that there's these two parts, right? The nociceptive part, which is just the feeling of pain, and then there's how much you care about the pain. There's the emotive part. And many of you might have had chronic pain and had some form of analgesia. And often what happens is you still feel the pain, but you care less about the pain. If you've ever had that experience, been in hospital, a lot of those drugs just work on your response to the pain. And those two things interact. So in psychology, a clinician will often work with a person to try and reduce that emotional content to the pain. So it's not taking away the nociception, but it's removing the affective or emotional content to try and reduce the experience of pain.
Jason, is that true that actually the brain doesn't really have the pain receptors in a way?
Exactly.
So I mean,
it's a peripheral.
It's a peripheral.
Nociception is peripheral. Yeah, exactly. And then the affective part is central.
This is complicated stuff, but beautiful. Lovely questions. All right, I've got one last question before we open it up to the audience. Modern neuroscience has been around for almost a century, but why are there not more treatments for neural disorders?
Well, look, I mean, there's my view, if you like. And if you think about, let's say cardiovascular disorders, high blood pressure, heart attacks and things like that, we understand a lot about the way the heart operates, what runs its, the way it beats, how fast it beats, and what controls it. And we know pretty well what happens after a stroke, a lot about how the muscles die, how they recover. And the treatments that have come out of that have come basically directed following that understanding, more commonly than not after animal models. Now, in terms of brain disorders, I would say the vast majority of them, we barely understand what's going on. And even then, we don't really understand where you would start to treat it. That's one problem. If you can find a drug that might work, getting it into the brain is not straightforward. There's a thing called the blood-brain barrier, which separates your body from your brain, and getting things across there are difficult as well. And Parkinson's is a classic example of this. We've known about exactly what's dying in Parkinson's disease for probably 50 years now. We know exactly which cells are dying. We know exactly how many are dying. But in terms of fixing Parkinson's disease, we still use the same drugs we did when I was a medical student. It has not changed.
Wow.
So, and that's really because we don't know why those cells are dying. And that's the crux of the problem, which is why places like the QBI exist.
There you go. Do either of you two want to have a go at this as well?
Yeah, I mean, I think following from Pankaj, it's the same situation with respect to anxiety and depression kind of disorders, where again, I think even though we have improved our understanding, that understanding has led to the view that actually these disorders are quite heterogeneous, and they're not just located in one area of the brain, unlike the Parkinson's, where, we can define that these are certain type of neurons which are dying. But for example, when you're subjected to chronic stress, there are a lot of connections in the brain that get miswired or misconnected, and trying to fix that or imbalance in different neurotransmitters, and trying to fix that at one place actually creates an imbalance at the other place. And that's why some of the, the traditional very old barbaric sort of techniques that existed probably in 1940s, the lobotomy kind of experiments or kind of surgeries that were conducted where they inserted needles in people's brain and just moved needles in direction, trying to actually just sever some connections. And that gave some interesting results initially, and it was something that was performed and made popular in 1940s and 50s.
Win a Nobel Prize.
Yes, Igaz Moniz, the Portuguese neurologist, actually won a Nobel Prize for sort of inventing that technique. But later on, as it turns out, that this was really just a shot in the dark. Really didn't know what he was severing, which regions were affected, and as it turns out, there were many cases which just went bad in terms of, there were a lot of other issues. People actually experienced apathy. They couldn't feel emotions anymore. And that's where I think it stopped, because that's when the advent of antipsychotics and antidepressants came around in 1950s, which again, interestingly, were serendipitous discoveries. I mean, these drugs were developed to treat tuberculosis, and they just observed that those patients were feeling fine and happy. And that's how this whole, the area of antidepressants took over. So we still really don't understand how these antidepressants actually work in the brain, and what are the different processes it can actually regulate.
This stuff is unbelievably complicated, but also unbelievably intriguing. We now come to questions from the audience. Has anyone got a question that they would like to pose? We've got some hands over here. The microphone will come to you. If you could wait until you've got it, your words will be recorded for posterity as well.
Yes. memory is you know, well, so many fascinating things here. It's hard to pick which to lob a question into. But I years ago read that that memory was like a hologram. I'm a physicist, and you can take these pictures that are actually stored on a little slide that doesn't look like anything when you look at it. You know, ordinarily you can see a slide, you see a picture, you see people. And yet in the brain, it seems that the memories are not located in each neuron. In fact, what you're talking about, what you've been talking about, is new to me. The idea that it's not in the neurons, it's in the connections, and the strength of the connections where the memory is, which seems at odds with the AI, at least if you're using computers, where the memory is in the chip, is in a state up and down, and the connections are just wires, sort of thing.
But you can
anyway,
think of those connections as being, you know, they're like the synapses, right? So you couldn't point to a part on a chip and say that's the memory of the essay I just wrote. It still waits in a network, and we think that's how the brain does memory as well. So I think they're kind of, algorithmically, they're very similar, the way memory works.
Maybe that's why AI can get along a bit and do the things it does. But the question that I want to ask is, with what Denisha was saying about the new neurons, the new parts of the brain that grow, contrasting this with the old memories, like, you know, I always think about my primary school. I got a memory about my primary school still, amazingly enough, and that will be stored in my brain. But with these new neurons and the new connections they make, do they then participate in holding that memory, or are they all for, you know, learning the streets of London before you get your taxi driver licence? How do they fit into the whole thing?
That's a really interesting question, and I think what we know is perhaps these neurons are not participating in the memory that has been already formed, but rather these neurons are very important during learning processes. And it allows a level of flexibility, so as to say, in terms of, you know, whether today you parked your car in a particular bay versus the other bay. I mean, to remember where exactly you have done that, and to separate those, you know, it was not bay number 22, but bay number 35, probably requires these aspects of new neurons, which can give the added flexibility. So I would say these new neurons are more involved in integrating information and during this flexibility aspects of memory.
We have another question here, I think.
It's about anorexia. Is it a topic that you're covering at all?
Yes, indeed. We actually we have a trial at the moment. We are stimulating people who are not responding to drugs, and trying to treat anorexia. It's very early days, though.
Did you have a specific question that you wanted to ask about that, or just whether we're working on it?
So we're looking at the therapeutic side of it, not at the reasons why people get anorexia. A treatment methodology, yeah.
Question at the back.
Yes, I'm just interested in progress on learning about developmental trauma and the impact of in infancy and the impact on brain development. And would it be fair to say that in infancy we don't have a language yet, and that everything we experience is a sensory experience that's stored in the nervous system, and if we experience something that's too overwhelming, then it would dysregulate the nervous system, impacting on behaviour as we develop?
Well, certainly early life experience. I mean, if you think about the brain is wiring itself up, so to speak, from very early on. So exposure in the environment, every interaction that a child has is going to influence the nature of that wiring up process. That makes sense. If we think about something very extreme, there's interesting research that was done many years ago looking at children who had intractable epilepsy, you know, from birth, And the only way to stop the seizures, these very disruptive seizures, is to take out large parts of the brain that are sort of the focus of that abnormal electrical activity. And in some really extreme cases, that could involve one whole side of the brain taken out in those individuals. And what's really remarkable is that scientists have followed up a number of those cases over the years and found that those individuals, despite having half of the cortex removed at a very early age, were able to adapt and live relatively normal lives later on. Only very, very subtle impairments later on in life. You could not do that in an adult. So very significant insults early in life can nevertheless be compensated for.
Any further questions?
Yeah, like my question actually follows on from that nicely. My program of research is neurosciences in children and infancy. And what we're currently thinking about and exploring is, the thought about neuroplasticity versus vulnerability. Because, the older school thinking about brain injury in infants and that they actually recover better has somewhat been debunked because you can only pick up some deficits. And I'm talking to the expert here as they kind of hit their milestones and they get their neuropsych assessment. And, specifically neurobehavioral problems that actually are quite impactful on not just the patients, but also the families. And then thinking about those, like what you were describing, those adverse childhood events and how that impacts the patient and the family. Is that an area of research that you focus on in terms of neuroplasticity and exploring, how those cells that work for adults, how they work for children, that, when they have a brain injury, don't just lose skills that they've already learned, but they're actually meant to pick up skills, during that time. They might be in hospital and they actually separate from their peers even further. Mm.
We don't have active research going on in that area.
There's no one
it's
in
a
the
very
QBI really
one.
who's working with kids with injury like that. Mm.
Mm. Yes.
Hello. I have way too many questions, but I'll just go for one. I've been very interested in the brain state, the default mode network, and I was wondering if you could speak to that.
I didn't quite understand what
the default mode network.
Oh, the default mode.
Default
yeah,
mode
interesting question. So this is, I guess Jason can talk about this as well. This is. Maybe you should.
Yeah, sure. So this network that's been asked about, if you have a person in an MRI scanner and you look at brain function while they're at rest, so not doing anything in particular, just at kind of quiet rest, there's a specific network of the brain that becomes very active, a set of connections, and that network can be predictive of a whole range of sort of day-to-day functions. And there's been a lot of work on what the relevance of the default mode network might be, why it differs between individuals, how it might change over time, and so on. Did you have a specific question about that network?
Thank you. I know that it goes to 4 different regions of the brain, and I was wondering what might be, like what happens in those regions of the brain. Why might the default mode network be present in these specific parts of the brain?
Nobody knows. No, it's an area of active investigation. Why would it be those 4 in particular? It might be to do with consolidation of learning experiences. So when you're not actively engaged in a task, in fact, when you're disengaged from all tasks, this might be an opportunity to consolidate memories, as Pankaj was talking about before. That's one hypothesized function, but there are many.
Okay.
Electroconvulsive therapy has been widely used to treat mental illnesses, including some discredited practices at Chelmsford Hospital in Sydney about 40 or 50 years ago. Just wondering what the current theories on ECT are today.
Well, look, ECT is very effective in some disorders, and it works for a lot of people, and some people it doesn't work for. Exactly what ECT is doing is not very clear at all. The theory really is that it alters the plasticity that's already present. So some of the disorders that are going on are due to plastic changes in the brain, and it's a reset. That's really the theory. As to why it is that things come back, which they often do after ECT, it's really not very clear at all. But for some forms of depression, certainly it seems to be very effective. And it's probably less used now than it used to be, particularly with the advent of more pharmaceutical treatments, but it is still widely used.
One more.
Sorry, I just have a question. Like, people think a lot or talk about meditation. So what is meditation doing to the brain?
You want the short version or the long version?
I mean, it's studied. This is a topic that is studied. There is neuroscience research on meditation and what happens in the brain. Obviously, I guess as a non-meditator myself, one of the points is to reach a kind of different state of consciousness, perhaps a kind of relaxed state of consciousness. And that raises the whole issue of what is consciousness, this idea of this kind of inner subjective life that we have. And there is a lot of scientific interest in that, but it's a really, really hard problem. It's certainly possible to put, for example, people have compared meditators and non-meditators, put them into an MRI scanner, look at patterns of brain activity, and there are reliable differences, but they don't really tell us why there are those differences and what they mean. So I think there's a long way to go to understand particularly this problem of consciousness and changes in people's subjective states of consciousness.
I think we're going to have to call the discussion to a close. What you've seen and heard today will give you an idea of why the brain is such a compelling topic of interest, how complex the problems are, and why people, like my colleagues on the right, are needed to have a look at them seriously. The vision of the QBI is improved lives through a deeper understanding of the brain in health and disease. That's a marvelous statement. It seems to me to capture exactly what they're about. If you'd like to pursue these things further, I strongly recommend their website. It's just QBI@UQ. You'll find it very easily. And especially under the Brain tab, you will find issues of this journal, which is online. You have on your seats at the moment a paper version of it, with up-to-date research on the brain written in ways that an intelligent, curious public can understand, which is quite a gift, actually. It's great stuff. Under Engage on the website, you'll find talks and the Brain Bee, which is a particular quiz thing for Australian school students about the brain. And under News, you'll find a series of podcasts. And listen up, people, a new series of podcasts goes live this Friday. It's very worthwhile keeping in touch with the ongoing movement of research in this area because, as you understand, it's fast, it's complex, and there's a great deal going on. The people at QBI take seriously not only the business of doing world standard research, but also sharing it with people like us, who may not be experts, but who have a strong interest in it. I think that's very important to modern science. It used to be the case that you could be someone squirreled away in a lab and not worry about the public at all. A responsible, big institution like this one is now committed very strongly to making sure that its work gets out there and is understood and helps to form people's understanding of what they're on about. And so our thanks to the experts today, Professor Pankaj Shah, Professor Jason Mattingly, and Dr. Danisha Javeri, and especially to you, the audience, both here and online, for attending to celebrate 20 years of the QBI at the University of Queensland and to look forward to its next 20 years and beyond. Professor Pankaj Shah, to you and your colleagues, a happy 20th birthday indeed. Thank you. There will now be tea, coffee, and associated nibbles, I think, out to my left, your right.